Okay, today we're going to do photosynthesis, part 2. Calvin cycle. Don't get it confused with the Krebs cycle. Calvin cycle is in the chloroplast. Krebs cycle is in the mitochondria. It's part of cell respiration. So speaking of photosynthesis, let's do a recap of what we did the last two classes real quick. We talked about part 1 of photosynthesis, which are the light-dependent reactions. I'm going to just call them the light reactions. It's really the same thing as light-dependent. In fact, it is the same thing except for the name. Light reactions are the energy capturing phase of photosynthesis. In part 1 of photosynthesis, you make the energy. In part 2, you use the energy. Remember that the location of the light reactions was the thylakoid membrane. It basically serves as an electron transport chain. The inputs, the reactants, what you need, it's going to be water, sunlight, ADP with an inorganic phosphate. They're not attached yet. And NADP+, that is your electron carrier in its oxidized form. The outputs are going to be oxygen, adenosine triphosphate, ATP, and NADPH. What does a plant generally do with the oxygen that it produces? It goes out of the plant. How does it get out of the plant? What are those openings in the bottom of a leaf called? Stoma. So this is going to go back to the atmosphere, and it's going to be used by us and animals and every other creature that requires oxygen. Where are the ATP and NADPH going? Think of your diagram from Friday. They're going to the Calvin cycle. They're going to the stroma. All right. What reactant from photosynthesis do you not see here? What is something that we write in the equation for photosynthesis that has not been used yet? Right? Glucose is not a reactant. Glucose is a product. It's made in photosynthesis. Jasmine? Carbon dioxide. We have not mentioned carbon dioxide once until today. Now we're going to get into it. So, Calvin cycle. This is the energy harvesting phase of photosynthesis. You finally made your energy. Now you're going to use that energy to make sugar. Location is the stroma, which is the open area within a chloroplast. The inputs: carbon dioxide, ATP, and NADPH. How did CO2 get into the leaf? Same way oxygen came out, through where? The stoma. The outputs are going to be C6H12O6, adenosine diphosphate with an inorganic phosphate, and NADP+. Where are ADP and NADP+ going? Where? Yeah, more specific, they're going back to the light reactions, which is where the electron transport chain is. They're going back to the light reactions to get reloaded. Same reason why you got to go to the gas station, reload. Same reason why you go to the grocery store, reload. Okay. So the Calvin cycle can be divided into 3 stages. So what I'm going to do with you guys is I'm going to name these stages, and then we're going to diagram them. We're going to make a drawing, sort of similar to what we did with the Krebs cycle a couple weeks ago, or actually it was last, I think, Monday or Tuesday. And then we're going to describe in words what's happening with each stage, and we'll get those notes written down. So the stages are as follows. Stage 1 is called carbon fixation. Stage 2 is energy investment. Step 3 is regeneration of REBP. All right. So let's actually get into drawing the Calvin cycle. I think you're going to need 1, 2, 3 or 4 colors. Nothing major. I want you to remember that when I draw a little red circle, that represents a single carbon atom. How many carbons are there in glucose? How many carbons are there in carbon dioxide? Look up at the whiteboard. How many carbons are there in ribulose bisphosphate? How about PGA? And how about G3P? So we have a 5-carbon molecule, a 1-carbon molecule, and we have 2 different types of 3-carbon molecules. Let's get this party started. Here we go. So the first thing that happens is you have 3 carbon dioxides. 1, 2, 3. They are going to enter the Calvin cycle. Each carbon dioxide has 1 carbon, so you have 3 separate carbon atoms. They are going to interact with 3 molecules that have 5 carbons each. And on both sides of this molecule, there's a phosphate group. So this molecule has a proper name, it has a nickname. The proper name is ribulose bisphosphate. Its nickname is RUBP, and it is a C5 molecule. You have 3 of them. Lydia, where are you? Okay, guys, this is really important for you to keep track of the numbers here. The numbers don't lie. Everything's going to add up. there's a lot of weird names, but you guys are AP students. Don't let the names intimidate you. It's just going to be simple chemistry. The fact that this ends with the you don't need to do this highlighting. It ends with OSE. What does that mean? It's a sugar. Okay. And the fact that you have the prefix bi, what does that mean? Two what? Two phosphates. If you're wondering why there's this, sometimes the S is there just to connect the words. So we have 3 molecules that have 5 carbons each. They're going to join with 3 carbon dioxides. And what they do is that they actually are going to be used by an enzyme. This enzyme is called Rubisco. Oops. Rubisco is the short name. I'll give you the real name. The enzyme is called ribulose bisphosphate carboxylase. What part of that word lets you know that it is an enzyme? ACE. Its nickname or abbreviated name is Rubisco. Ru for ribulose, bis for bisphosphate, and co for carboxyl. This enzyme allows for the 5 carbon RUBP to join up with the 1 carbon CO2. It allows them to come together. In fact, carboxylase means like to add carbon. You're going to add a carbon. So what is 5 plus 1, Jet? That's it. So we are very, very, very temporarily going to have 3 6-carbon molecules. This is a very short-lived molecule. So we have 3 C6 molecules. This molecule, you don't even need to bother with the name because it is very short-lived. It doesn't last long. What I mean by it doesn't last long is that the molecules are too big, they're going to break in half. That's what we're going to demonstrate here. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. Oops. This molecule has a name. It's called phosphoglyceric acid. You can call it PGA for short. And there are 6 of them, so you have 6 C3 molecules. And just to prove to you guys that the numbers are adding up, I want you to look here. How many total carbon atoms do you see? Total carbon atoms. 18. How many do you see here? 18. It just got too big in a split. Is everybody okay so far? Have I lost anyone? All right. So next step, I want to be very clear about something. This is where you're going to have ATP and the NADPH, but I need to make this very clear. You ready? Just read before you write. Don't give yourself a break. I want you to hear this before you write this. Can't miss this point. Each PGA will be phosphorylated by 1 ATP and reduced by 1 NADPH. So, here's what I'm getting at. Guys, how many PGAs do you see there? 6. Each of them is going to be phosphorylated by an ATP. Each. So how many ATP will we need? 6. Each of them is going to be reduced by 1 NADPH. So how many NADPHs do we need? That's my point. Each of them is going to get an ATP, and each of them is going to get an NADPH. So we need 6 of each. So rather than draw 6 ATPs and 6 NADPHs, I'm just going to say 6. 6 ATPs are going to jump in here, and they're going to react with the PGAs. That's going to result in the ATP becoming 6-ADP plus 6 inorganic phosphates. And we're going to have 6 NADPHs that's going to result in 6 NADP pluses. So they're going to phosphorylate and reduce the molecules, and that is going to form new carbon molecules. These molecules are ones that you have heard of before. They're called glyceraldehyde 3-phosphate, or G3P. Some of you may want to go into science when you go to college, and if you want to do that, like be a doctor or a biologist or biotech or anything like that, you might have to take organic chemistry. And in organic chemistry, you learn how to make and name these organic molecules. And what I'm getting at is this name, glyceraldehyde 3-phosphate, that 3 does not mean that there are 3 phosphates. That 3 means that the phosphate group is on the 3rd carbon. In organic chemistry names, you sometimes have to identify which carbon has an additive ion or element to it. So glyceraldehyde is the name. The 3 means the 3rd carbon in the chain, and the phosphate means that that phosphate is hooked up to the 3rd carbon in the chain. That's what that means. And you don't really need to know that for this class, but if you never knew, that's what those like odd names with the words and the numbers come from. Those organic chem names. Yes, Ayla?
So is it like for every 6 like reactions or over how many ATPs you produce, you get 1 Calvin cycle reaction, or is it 1 after the other?
I don't understand what you're asking.
So you know how you need 6 ATPs and 6 NADPs?
Yes.
So do you make 6 from the like reaction?
Well, every 1 water molecule makes 1 NADPH. So you need 6 water molecules for that. And you're just pumping a lot of hydrogen into the lumen, so you're making a lot of ATP no matter what. But the number of NADPHs you make is dependent on how much water you break down.
So are you making exactly 6 then?
You have more than 6, but you need exactly 6 to make this work.
So there's an excess of ATP?
There's definitely an excess of ATP. Not as much so for NADPH. Yeah. Each PGA will be phosphorylated by 1 ATP and reduced by 1 NADPH. That's why there are 6. All right. Now, guys, one of these, let's highlight one. One of these G3Ps, just one, is going to leave the cycle. This is going to contribute to making 1/2 of glucose. Just by using your own eyes, how many carbons are there in a G3P molecule? There are 3 of them. How many carbons are there in a glucose molecule? There are 6. You need 2 G3Ps, so you have to do the whole Calvin cycle twice. You have to go through the entire Calvin cycle 2 times to make 1 glucose. Now, someone earlier today asked, why don't you just take 2 G3Ps from this? You're going to find out why. If you consider the carbons that are not highlighted, how many carbon atoms are there that are not highlighted? 15 carbon atoms. Look up here. We have to make that again. How many carbon atoms are there? So let's see if you can answer the question that a student asked earlier. What if, and don't do this, don't highlight, but just pretend. What if I, instead of using 1 G3P to make glucose, I use 2 G3Ps? Problem solved, right? Glucose is 6 carbons. I'm just going to use 2 of those to make glucose. 3 plus 3 is 6. So how many carbons do I have left over? How many total carbon atoms? Just count. 12. How many carbon atoms do I need to make 3 RUBPs again? 15. You're not going to have enough. So you can't do 2 G3Ps. You have to just go through the Calvin cycle 2 times. So we're going to go up here. And in order to regenerate G3P, we're going to need 3 more ATP. And that will form 3 ADP plus 3 inorganic phosphates. And that's going to help the regeneration of RUBP, ribulose bisphosphate. Okay. So now that we have the entire Calvin cycle drawn, I'm going to break it up into the 3 stages, okay? Here we go. Between here and here will be stage 1. So when we're writing these notes that we're about to do, you can refer back to this diagram and identify which part I'm referring to. Okay, next. This is 2, and then there's 3. Those are the 3 stages of the Calvin cycle. And what we're going to do now is we're going to describe what is happening in each of those stages. All right, so let's do that.
Wait, what is the, did you say after RUBP, just 3 minus C6?
Where are you looking at, Jet?
The first section.
No, I'm just saying that there's 3 C5s. There's 3 C6s. There's 3 C6s, or 3 C6s, 6 C3s. I'm just telling you the number of the carbon atom, or the molecule. Okay, here we go. Let's go over what's happening in each of them. Step 1, carbon fixation. In carbon fixation, atmospheric CO2 is fixed, and whenever you see the word fixed, it typically means captured. It is captured by an enzyme called ribulose bisphosphate carboxylase. Its abbreviated name is Rubisco. Rubisco. Ribulose biscarboxylase. So what you all have is you have ribulose bisphosphate. You can abbreviate that as RUBP. You remember my rule. You're not allowed to abbreviate unless you know what it means. If I find Dan in the hallway and I ask him, Dan, what does RUBP stand for? I don't know. You're not allowed to use the abbreviation. That's the condition. You have to know what it stands for if you're going to use it. RUBP is a C5 chain. It is a 5-carbon chain. Adding CO2 makes a temporary C6 chain. The 3 molecules of C6 break up into 6 molecules of C3 called phosphoglyceric acid. You can just call it PGA. PGA will be fine. If you wanted to use that on your FRQs, that's fine. Just please be sure you know what it stands for. So bring it all together. Three RUBP plus 3 CO2 yield 6 PGA. That's basically it. That's what you're doing in the wrong one. That's what you're doing in the first part of the Calvin cycle called carbon fixation. Okay. We have about 10 minutes left, so let's get moving. Part 2, energy investment. In the energy investment stage, listen up. Each PGA molecule is phosphorylated by 1 ATP and reduced by 1 NADPH. This results in 6 molecules of glyceraldehyde, 3 phosphate. This is called G3P. So you basically have 6 PGA plus 6 ATP plus 6 NADPH results in 6 molecules of G3P. But there's a condition here, guys. One of the 6 G3Ps leaves the cycle to contribute to glucose. As I've already said, each G3P has 3 carbons. A glucose has 6 carbons. So you must go through the Calvin cycle 2 full times to make just one glucose molecule. Then we get to the third and final stage, the regeneration of RUBP. The 5 remaining G3P are phosphorylated by 3 ATP and after a series of reactions that you guys don't need to know about, RUBP is regenerated. So we have 5G3P plus 3 adenosine triphosphates results in 3 RUBP again. And you have completed the cycle. So I have an animation here that I want you to see. If you're watching this on the video, I'm not sure how it's going to convert. Hopefully it's not going to be too jumpy where it looks like a frozen image. We'll just have to wait and see. Just 1 second. Okay, perfect. So let me share the screen, make sure we're sharing the right thing. All right, cool. I would like you guys to watch this screen. It's about as big as I can make it without cutting out a part of it. So if you look on the left, I'm going to talk loud. If you look on the left, you see that there are 3 molecules of RUBP. This is Rubisco, your enzyme. Here's your 3 molecules of carbon dioxide. We're going to do a head count for how much ATP that you use. Stage 1, Rubisco attaches the carbon to RUBP. This is known as carbon fixation. All that's going to happen is that this is going to attach to each of these, thanks to the enzyme, and it's going to form 6 molecules of PGA. Here it goes. 2, 4, 6. So did you use any ATP with that? How about any ADPH? No. The enzyme just brought them together, and the molecule was so big, it was a 6-carbon molecule. It broke apart. That was phase 1. 2, you're going to turn PGA into G3P. So you used up 6 ATP, 6 ADPH. One of the 6 G3Ps you make is going to go to help make glucose. Yes, Dan?
What's the point of the ATP adding a phosphate if the NADPH is going to take it away?
It's not the same phosphate, and that's going to be part of the it's going to oxidize. And when you're oxidized, the electrons and the protons are going to be attracted to each other. The phosphate was attached to one side of the molecule. the phosphate that's taken away was a different phosphate. It was one that was already there. So you added one phosphate already. You took away a different phosphate that was in there. So it's the location of the phosphates really does dictate the behavior. You know, if I took away your right shoe, that's not the same as taking away your left shoe. All right, here's the third stage. The regeneration of RUBP. Okay, so let's go back to your notes, finish them off. Products of the Calvin cycle. 1G3P. That's going to be half of glucose. 9 ADPs. You used up 9 ATPs, so now you're going to form 9 ADPs. They're going to go back to the light reactions. They're going to reload. And you're going to form 9 NADP pluses. They're going to go back to the light reactions and reload on electrons. If you have one G3P plus another G3P, you will get glucose. So how many times, again, do you have to go through the Calvin cycle to make one glucose? You have to go through the Calvin cycle twice. Now, I told Ayla a little while ago, for every NADPH that you make, you have to break down one water. How many NADPHs do we use in that whole cycle just now? Did you catch that number? 6. So how many water molecules do you need in order to go through photosynthesis? You need 6. So to get this whole thing going, you need 6 water molecules. But hold up. You need 6 water molecules to go through the Calvin cycle once. So how many water molecules do we need it all together to make one glucose? 12. You needed 3 carbon dioxides to go through the cycle once. You're going to need 6 to go through it twice. You make C6H12O6. You made 6 molecules of O2. And when you go through the third step, guys, that's a dehydration reaction. In a dehydration reaction, you take water out to bring molecules together. And so this is going to cause you to make 6 waters. But for those of you that have taken chem, when you see the same reactant, when you see the same substance on the reactant side and the product side, what do you do with them? You cancel them out. So this is going to get canceled out, and this 12 gets reduced to 6. And that's the actual equation for photosynthesis. We'll recap this on the next class, and then we will do different types of photosynthesis tomorrow.
What's the lattice?
Hold on, Jet.